Meiosis is a process where a single cell divided twice to produce four cells containing the half of the original amount of genetic information.
Have you ever wondered how two siblings can have mom and dad so different?
Now, today, we're going to talk about the processes of meiosis, which is similar to mitosis.
Mitosis makes identical body cells like your skin cells and stomach cells.
Recall from a mitosis clip that since it makes identical body cell, mitosis is important.
For growth and for repair of damage or to replace worn out cells.
But not meiosis. The process results in four daughter cells that are haploid, which means they contain half the number of chromosomes of the diploid parent cell.
Meiosis has both similarities and differences from mitosis, which is a cell division process in which a parent cell produces two identical daughter cells.
Meiosis is a process that contributes to genetic variety.
This process is required to produce egg and sperm cells for sexual reproduction. During reproduction, when the sperm and egg unite to form a single cell, the number of chromosomes is restored in the offspring.
During the S phase of interphase, the DNA is replicated, producing two copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis.
Meiosis has both similarities to and difference from mitosis, which is a cell division process in which a parent cell produces two identical daughter cells. Meiosis begins following one round of DNA replication in cells in the male or female sex organs. The process is split into meiosis 1 and meiosis 2, and both meiotic divisions have multiple phases. Meiosis 1 is a type of cell division unique to germ cells, while meiosis 2 is similar to mitosis.
Meiosis 1, the first meiotic division, begins with prophase 1. During prophase 1, the complex of DNA and protein known as chromatin condenses to form chromosomes. The pairs of replicated chromosomes are known as sister chromatids, and they remain joined at a central point called the centromere. A large structure called the meiotic spindle also forms from long proteins called microtubules on each side, or pole, of the cell. Between prophase 1 and metaphase 1, the pairs of homologous chromosome form tetrads. Within the tetrad, any pair of chromatid arms can overlap and fuse in a process called crossing over or recombination.
Chromosomal material is exchanged between the two pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no two siblings, aside from twins, are genetically identical. There are several more key steps in prophase 1. The nuclear membrane begins to break down. Then, the two centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of meiosis 1 is called metaphase 1. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is anaphase 1. During this phase, homologous chromosomes separate and migrate to the two poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis 1 are telophase 1 and cytokinesis. Here, the cell divides into two daughter cells.
Meiosis 2 is a mitotic division of each of the haploid cells produced in the meiosis 1. During prophase 2, the chromosomes condense and a new set of spindle fibers forms. The chromosomes begin moving toward the equator of the cells. During metaphase 2, the centromeres of the paired chromatids align along the equatorial plate in both cells. Then in anaphase 2, the chromosomes separate at the centromeres and the spindle fibers pull the separated chromosomes towards each pole of the
In
cell.
telophase 2, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis 2 began with 2 cells, and each of those cells were split into 2 cells, we now have 4 unique haploid cells at the end of meiosis. These cells are gametes. 2 gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.